An important issue in genomics right now is that we know what the genes are in the genome, but we don't necessarily understand what most of those genes are doing. The goal of functional genomics is to figure out what the genes are doing in the cell. By sequencing leiomyosarcoma samples, for instance, we may identify some abnormal genes because they are gone or mutated in the cell, but many of those mutations are what we call passenger mutations, just along for the ride with driver mutations, which are the real problem. Also, there's many ways to modify gene function other than mutation. So functional genomics helps us identify which genes and mutations are important and which ones are irrelevant for the cancer cells. The simplest functional genetic experiment is to turn off one gene and see what the consequences are for the cell. If I block a gene in leiomyosarcoma cells and all of them die, that gene must be really important for leiomyosarcoma biology. We can now perform large-scale experiments in which we block each and every gene of the genome individually. That's about 20,000 genes that can be manipulated individually. Thanks to technologies developed between Harvard and MIT and the Broad Institute, we have libraries of reagents that allow us to block each and every gene in our cancer models. This has never been done before in leiomyosarcoma. It wasn't even possible. But we've been working hard in recent months to improve our models and to adapt this functional genomic platform to perform experiments that we're confident are going to provide very useful insights into leiomyosarcoma biology. Functional genomics does not replace genomic sequencing. These are complementary approaches. There are several functional genomic tools available, and we use them for different purposes. It's a very exciting time in the field. CRISPR libraries are a very robust technique for screening purposes. They give a very clean result with a strong signal, but they have limitations, for example, for genes in amplified regions. So we find RNAi tools still very useful. shRNA libraries provide information that may be more relevant for therapeutic discovery. Certainly, the combination of both modalities provides the most comprehensive picture, and we are fortunate enough to have access to all of them through established collaborations. As soon as we identify genes that are essential for leiomyosarcoma biology, we'll immediately test any pharmacological agents that may modulate that gene or its pathway in the leiomyosarcoma cells that we have grown in the lab to validate this gene or pathway as a therapeutic target. In this project, we'll be performing two sets of experiments side by side. First, to identify genes that are essential for leiomyosarcoma cells proliferation and survival, we'll run experiments with patient-derived cell line models that we've established in the lab, which are representative of the clinical reality. But we already know that leiomyosarcoma cells are very dependent on PI3K signaling, and there are pharmacologic inhibitors of this pathway that have provided some, although limited, clinical activity. So we want to interrogate cells treated with PI3K inhibitors to see if there's anything else we can do to maximize the effect of these drugs, either attacking a second node or along the same pathway maximizing the effects of PI3K inhibition. Both of these arms complement each other and combined will allow us to design rational and effective therapeutic strategies for leiomyosarcoma patients.